is Rethik Sir, your biology SME. Welcome to the MemoNeet, one of the best app for your NEET preparations. Today we will be discussing about the law of dominance, okay, and law of segregation and different laws that comes under it. See, when I'm talking about a law of dominance, previous video we have actually discussed about how the characters have been expressed, right? So taking tall and draft characters as an example, or white and purple or white and violet flowering plants as an example. So based on that, we understood there is some factor which is okay. So there is a particular character which is actually characters that have been controlled by some specific factor or something. Okay. So then there has to be, even though we know that there are two different characters, but after breeding or after when they were being pollinated, okay. So what happened as a result? In next generation, the dominant one, I mean, out of those two character, only one character has been expressed or it has been expressing. So this says that there might be some character which is showing dominancy over the other. That means the character which is unable to express, we called that character as recessive one. And the character which has been expressing, we call it as the dominant one. So the characters are controlled by a distinct units. We named those units, not we. So what happened? The Mendel has named the unit as factor. So factors occurs in pair. That means whenever there is a factor, it normally occurs in pair. Say like TT and small t. So these are pair. So what happening here? The factors are always occurring in pair. In a dissimilar pair. Dissimilar pair matlab, so it could be capital T and small t. Here it is dissimilar pair of factor. One of the member here out of these two, one of them, okay, one of the member of these two pair in between these two. So one will be dominant and the other will be what? Recessive. Where one will be dominant. So that is what law of dominance states. So the law of dominance is used to explain the expression of only one of the parental character. Okay, that is especially when I'm talking about the monohybrid cross in F1 generation, we can see this law of dominance. Okay, and the expression in both in the F2 generation, there also we will be seeing the law of dominance where it explains the proportional, that is 3 is to 1 and obtaining in the F2 generation. Okay. So that is 3 is to 1 where 3 will be the taller plant and 1 is the draft plant, the ratio. Okay. And where it has been obtained this 3 is to 1 ratio in F2 generation. Okay. But now even understanding, consider this law will not be applicable in everywhere. Clear to you? This law is not applicable in everywhere. How and why? See, when we see the law of segregation, the law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such an F2 generation, though one of this is not seen in F1 generation. We need to understand the statement here. The statement says that in law of segregation, say like what they did is again they have started experimenting in different flowers, different characters. So law of dominance was not same in all the experiments. A new result came and they suggested, okay, there might be something else phenomenon or mechanism. So the law of segregation states that the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, through one of these is not seen at F1 generation. That means, see what happens is in F2 generation, whatever the characteristics that we have been seeing in the parental, okay, whatever it might be of parental flower or plant or animal, whatever we have taken, So that gets recovered in F2 generation. I mean, if you take a tall plant and a draft plant, okay. So in F1 generation, what is happening now? So the plant that comes, right, every plant will be tall. But in F2 generation, when you do a selfing, okay, when you do a selfing here, as a result, what you're going to get? One is tall and tall, tall and draft, tall and draft, and draft and draft. So out of this, three are tall and another one is draft. So you can see this. So it is homozygous, this is homozygous, and this both are heterozygous. This is homozygous tall plant, this is heterozygous tall plant, and this is again homozygous draft plant. So in F2 generation, in F1 generation, what happened? Only the taller characteristic was expressed. But in F2 generation, both the characters of the parents was recovered. But this is not same in the law of segregation. What is happening here? Instead of getting any one that is dominant character, instead of getting expressed of any dominant character, here we are getting some new character, some new or other character. But in F2 generation, again, it has been recovered. The parental character has been recovered. That is what they're trying to say in law of segregation. Okay. See, through the parent contains two allele. Okay. Through the parents contain, sorry, two alleles during the gamete formation. The factors or alleles of a pair segregates from each other such that the gamete receives only one of the two factor. So it only receives only one of the two factors. Of course, the homozygous parent produce all the gametes that are similar with the heterozygous one produces two kind of gametes, each having one allele with the equal proportion. That is what I was saying here. So we will see it how and all. We will go with an example. So in law of segregation, you will see two different examples. One is you will see in an incomplete dominance, and another one you are going to see in codominance. Okay, so yeah, we will see it one by one. Okay, in the incomplete dominance, yeah, in the incomplete dominance, if we see here, so one example which was being taken was the coloration of the flower. So the flower which was taken was a dark flower, or it is also called as Snapdragon. Okay, so this is one of the good example to understand this incomplete dominance. Okay, so here what they have did is they have taken a dog flower or a snapdragon flower. So where they have in the phenotype, that is in the parental generation, they have taken two homozygous red and a homozygous white flower. Okay, so they are homozygous here. So one was homozygous red flower and the another was homozygous white flower. And they made a cross between these two flower. So when the gametes, so what will be the resultant gametes which is going to produce? Obviously, one will be the R and the another one will be the small r. So when these gametes were crossed with each other, okay, as a result, now what could be the expectational result according to law of dominance, red flower. But here, something new kind of flower color was appeared. Okay. After in F1 generation, what happened? They observed, yeah. So in this F1 generation, they observed a new character of flower that is pink color. Okay. So they observed what? Pink colored flower. That means instead of expressing any one character from the parent, okay. So what happened here? Instead of that, a new character was expressed. So that is the pink color was been expressed. So what I can say here is this capital R, okay, that is the red color character. So here it is, this is not completely dominant over the small r, that is white color character. Okay, so that might be one possibilities. But that means it is showing against and Mendel's law. In Mendel's law, what happens? The dominancy plays a very important role. That is what Mendel say. But it is showing something against the, it is not following the Mendel's law. That is monohybrid crossing. It is not following that monohybrid crossing. But when comes to its phenotypic ratio, it has changed from 3 is to 1 dominant. Okay. And the recessive ratio, dominant and recessive ratio, that the way it is showing is again completely different here. How and why? See, after getting this pink color, again what they did is they again made a selfing. That means so self-pollination was been done. As a result, in F2 generation, okay, in the F2 generation, so what will be the gametes now? Capital R and small r, capital R and small r. So now these are the two gametes, right? When crossing is been done here, so it will become capital R, then capital R, capital R, small r and small r. As a result now, what we are going to get? So it will be red in color. These two will be heterozygous, which is pink in color, and this is white in color. So now in F1 generation, what happened? The parental characteristic was not characteristics were not absorbed, but in F2 generation, the characteristics were recovered. So now we are able to observe both red color and white color flower with that additional pink color flower. So one thing that I can say that this capital R is not completely dominant over the small r. This is one point. So as a result, we are getting some other character that is pink color flower. So in F2 generation, when crossing was done, as a result, what we are getting? So we are getting red color, two pink color, and white color. So according to the monohybrid cross, what was our ratio? 3 is to 1 was our ratio. Getting what was our ratio? The phenotypic ratio should always be 3 is to 1 according to Mendele's law. Okay, where 3 must be the dominant and 1 ratio, whatever that is, must be the recessive one. But what is happening here? R is not completely dominant over the small r, and it made a possible, okay, to distinguish the pink color from the red color and white color. As a result, whatever the pink color we are getting, easily I can say it is an heterozygous, where red color and white color is an homozygous. So that is making easy to understand here. Okay, so what might be the phenotypic ratio then? 1 is to 2 is to 1. And genotypic ratio also becomes 1 is to 2 is to 1. Okay, so phenotypic ratio will always be a kind of, okay, what is the coloration, physical appearance? Where genotypic ratio is always about, okay, how many homozygous, heterozygous, and homozygous. So in both the way, genotype and phenotypic ratio are same. Where in incomplete dominance. So this is an example for incomplete dominance. Right. Yeah, that is what they are actually trying to say. So later what happened? Research, repeated Mendeleev experiment and various expression, that is whatever that I have observed, right? An intermediate progeny is nothing but a pink color flower was been produced. That is what they're explaining. Okay, moving to the next topic here. In the same way, so according to the genetics, we know that the dominancy is a relationship between the allele of one gene. In order to understand the concept of a dominance of alleles or characters, we need to know more about exactly what is gene. So far, we know that the genes are one which actually carry the hereditary unit or characteristics in a living organisms, which exist as actually what? Pair of alleles or pair of characters in a diploid organism. Say like if human being XX and XY. So what happens from parents will get again, it will be followed by X and X or X and Y. So these pair of alleles may or may not be similar. That's what I'm saying here. That is, heterozygous gene has two dissimilar pairs. Heterozygous where genes will be obviously two dissimilar pairs of allele. While in homozygous, both the gene will be identical to each other. Okay, both the gene will be identical to each other. But what if there is any, okay, if there is any mutation has happened or even a different changes, okay, that has undergone in the gene? Furthermore, what could be the outcome? That is what we need to understand. That means, so during the transformation, sorry, during the transfer of these genes from one generation to another generation, if there is any slight changes that could that will happen, okay, in between any one of the gene, yeah, or in the pair of gene, if any one of the gene has undergone through some changes, it could be either mutation or anything, okay. So what might be happen? Say like we will take an example of uh, a kind of gene that contains the information for producing certain enzyme, producing an enzyme. So now there are two copies of genes, right? Obviously, genes will be in a pair. So in out of these, okay, the two allelic forms in which one of the gene has gone through mutation or some changes. So now as a result, what happens? The normal allele, okay, the normal allele are going to produce what? A normal enzyme, okay, that needs to form the transformation of the transformation of the substrate. Yes. So what we can understand that the normal allele or an okay, so normal allele is going to produce what? A normal enzyme here. So that is needed for the transformation of a substrate. Yes. So therefore, if the gene has been modified, the other pair or other allele has been modified, okay, so that is going to represent the production of, yeah, so that is going to represent the production of these three ways. So one is the modified allele could be responsible for the production of either normal or less effective enzymes, which is going to help, which is needed for the transformation of the substrate, yes, or a non-functional enzyme or no enzymes at all, right? See, in first case, what happens? If the modified allele is equivalent to the unmodified allele, that means if the modified allele is equivalent to the unmodified allele, if it is showing the same thing, then as a result, what happens? It will be producing the same phenotype or trait. That is, it is going to produce a normal, okay, a normal enzyme, which results helps in the transformation of substrate. Yes. So such equivalent allele pair are very common. But if the allele produce a non-functional enzyme here, so if it produces what? A non-functional enzymes or a allele undergo mutation and forms a non-producing functional enzyme or no enzymes at all. So what might be the phenotype? So the phenotype might be affected now. Okay. So the resultant ratio might be affected now. So the phenotype or a trait, oh, that is nothing but character, will only be dependent on the function of the unmodified allele. So what exactly the allele is, whether it is modified or unmodified. Based on that, the phenotype will be. See what happens when it is unmodified allele or functional allele. Then as a result, what you'll get, you will get the original phenotype. That is, you will get that is the dominant allele and the modified allele is generally. So what happens? The dominant allele, right? It will generally, it is going to dominate over the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzymes are produced. So, based on sometimes the character is purely based on what? Purely based on alleles. That is what they are trying to say. If it is non-functional or completely recessive allele, the dominant allele is going to dominate over the recessive allele. But if the allele is non-functional, but it has gone through some mutation or else it is producing some other thing, then what happen as a result? So the phenotype also changes. Okay, so by this example, because it is going to help us to understand what exactly the dominant is and how exactly the dominant allele or dominant gene works. Clear? So this was all about what? Incomplete dominance. Next case, that is codominance. So what happens in codominance is, in case of codominance, still now we discussed that two cases can be happen. One is the dominant gene, right? Whatever the dominant character, it dominates over the recessive gene or recessive character. So that was one case. Second case, sometimes both will get dominant to, not the dominant gene won't get dominant over the recessive. So as a result, what happens? An intermediate character. Not that means not completely in incomplete dominance. The dominant character gene will not completely dominant over the recessive one. As a result, we will get an intermediate character or intermediate result. Whereas in codominance, what happens now? Here, both the okay. So in F1 generation, it resembles what? Both parents. That means even the father and mother. Or if there is some having parents are having different character. When crossing is done in F1 generation, whatever the progeny, right, it is going to show both the characters, okay, from the parents, sorry, of the parents. So one good example that we can take while studying this codominance is the RBC cell, that is red blood cells, okay, which determine what A, B, O blood grouping in human beings. Okay, and A, B, O blood group are controlled by gene capital I or gene I. So the plasma membrane of the red blood cells, it actually has a sugar polymer that protrude from its surface, and kind of sugar is controlled by the genes, that is capital I. So what happens here? So there is a table here. Just a minute. Yeah. So what happens? The plasma membrane, right, of the red blood cell, as I told, it has a sugar polymerase. It has what? A sugar polymerase that normally protrudes from its surface, right? So, and a kind of sugar has been controlled by gene I. That has also been said. So, gene I, it has actually three alleles now. There are, say, like three characters. So, one is IA, IB, IA, IB, and small I. So allele IA and IB, they produce slightly different form of sugar. They are going to produce slightly different forms of sugar. Okay. While the small I, it doesn't form any sugar at all. Okay. It does not produce any sugar because humans are, why? Because humans are diploid organism here. Since they are diploid organism, each person possesses only any two types of character out of this three I gene allele. That is, out of IA, IB, and small I, the human beings are diploid in nature, so they will be possessing only any two characters here. So out of these three, they will be having only two. Clear with it? So as a result, what happens when from a parent, and here we have what? Another parent. If IA and IB are completely dominant, okay, over the small I, and we need to understand here that these two, right? These two are completely dominant over the small I. But IA and IB, okay, when they are being brought together, right, they become completely dominant over each other. That means co-dominant. As a result, in F1 generation, both will be expressed. Say like it is an homozygous. So IA and another blood group, that is allele from parent one, that is IA. So allele from parent two, that is again IA. So what would be the genotype of the offspring now? So genotype would be IA and IA, that is homozygous in condition. As a result, you will get what type of blood group now? A blood group. But if the allele from the parent one is IA, and the allele from the parent 2, that is the character, the gene possessing the character will be IB, then as a result, what happens? Both the alleles are going to express together, okay, not an intermediate, together, exactly as what the parents consist the character. So as a result, IA and IEB are been expressed together. And what will be the blood group now? A and B. So it is having A blood group from parent one and B blood group. So the B character is from parent two. So as a result, it is having both the AB blood type of offspring has been produced. And I told you that IA and IB, IA and IB, right? So it acts as completely dominant over the small I. So as a result, what happens when IA and small I from parent 1 IB and parent 2 small I comes? So what would be the genotypic ratio? IA and small I, IB and small I. So what would be the blood type in the offspring now? A blood group. Here it will be the B blood group, right? So here it is A blood group. And obviously here we have B blood group. So and here again they are homozygous. As a result, it will be O blood group, universally donor. So O blood groups are universally donor. Okay. So what happens here? Where we are actually seeing the codominance here, we are actually seeing codominance in these two ways. That is I and IB, then same thing. That is IB and IB. So in these two cases, we have been seeing that both the characters of parents have been expressed in the offspring. That is, allele from parent 1 was IA and from the parent 2 was IB, or vice versa. As a result, what is happening? The blood type of the offspring is AB and AB. That means both the character of the parent has been expressed. So this is what codominance we call as. Okay. So totally how many combination of blood group can be studied here? 6 types of blood group combination can be seen, or it is possible here. Okay. So this was all about the codominance and blood grouping. Okay. Not only that, we can also study the multiple allele, taking this as an example. So multiple allele, how? So because here we can see that there are more than two, that is three alleles are being governed by the same character, right? So I is a gene and it is having different alleles, three different characters, which is being governed by same allele here. So if we take up the population, individual population survey and all, we will be able to find exactly, since an individual only will be having two alleles, But if we want to know the multiple alleles, we can actually, that can be found when a population survey has been done. Okay. Another way is, sorry, yeah, where it is, yeah. I'll take this then. Or else I'll take this itself. This is good. So occasionally what happen, if we take a gene produced, we know that a single gene will be producing more than one effect. For example, a starch synthesis in pea seed is being controlled by one gene. Okay. So that is the starch which is been synthesized by a pea seed is actually being controlled by one gene. It has one gene. So one gene has how many characters now? Two alleles. One is B and capital B and small b. So starch is synthesized effectively in dominant one, that is in the capital B and capital B, that too in an homozygous condition. And therefore, what happens? A large starch grains are produced. Okay. But in contrast, if it is homozygous recessive one, that is small b and small b, a least effective of starch synthesis can be seen. Okay. Produce a smaller starch grain. It is going to produce a grain, but the grains will be having what? Smaller starch in it. So after maturity, what happens? After the maturity of the seed, what happens? This capital B seeds are round. Okay. And small b, that is the homozygous recessive seed. Okay. So will be wrinkle in shape. This is clear. The homozygous capital B seed will be having more starch at it with the seed during after its maturity will be round in shape, where homozygous small b will be having seeds will be having less amount of starch and it will be wrinkled in shape. So in the heterozygous, what happened? They also produce a round seed because here dominancy comes. But we can see that capital B seems to be dominant over the small p, dominant allele. But starch grain production are intermediate size. That means, see, in where capital B and capital B, we are seeing what? The round with a very good amount of starch, where small b and small b we have been seeing a wrinkled seed with what kind? A wrinkled seed with less amount of starch. But if it is with capital B and small b, that is heterozygous in nature, so again we are being able to see what? A round color itself, but it is smaller in size. Okay. So intermediate size, that is smaller in size. So if the starch grain size is considered as a phenotype, then from this angle, the allele shows what? Incomplete dominance. Okay. Allele is showing what? Incomplete dominance here. So for incomplete dominance, you can also give this as an example. Okay. For incomplete dominance, we can also give this as an example. Therefore, dominance is not an autonomous feature of the gene. Like every time, dominance cannot be considered as a feature or feature of a gene or the product that has information for it. Okay, depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine. In case some more than one phenotype is influenced by the same gene. That means, see what happens is phenotype is every time phenotype has been influenced by same gene but having different character. So dominance cannot be considered every time that it is going to decide the phenotype of a gene. According to Mendelian monohybrid, we thought like, okay, dominance based on that, the phenotype has been judged. But here after knowing this experiments, we get to know every time the dominancy cannot be considered. Okay. Feature of the gene or the product that has the information for it. So it actually depends on what? It depends upon the much on the gene product and production of that particular phenotype from that. Okay. and a particular phenotype that we choose to examine. Based on that only, we will be able to understand the phenotype. Clear? So this was all about what? Law of independent assortment, sorry, incomplete dominance and codominance. Okay. So in this video, we actually understood that not every time dominant strategy comes, not every time the dominant character will dominate over the recessive one. Sometimes there might be an intermediate character can occur, or sometimes both the dominant and recessive, they might express together equally in the F1 generation. That is what we have studied, right? Taking a dog flower as an example and taking blood group an example, and for incomplete dominance, we also took the seed size and sugar production as an starch production as an example. So, more than this, if you still feel any doubts and all, you can comment in the message box and let us know in what way you are feeling difficulty. Meanwhile, very important announcement is maybe in coming Ganesh Chaturthi, again, we might announce some offers regarding the MemoNeet plan and all. So, even you can utilize during that time. So, we have Brahmastra test series because you still have just 7 months left for your NEET exam. So it's really very important that your preparation should start from now itself in a serious way. So one the best way is based on number of test taking the test. By taking test, you will be able to know exactly in what topics you are very strong and how the preparation, all our preparation is going on according to you guys. So for that, giving test is really very important, and yeah, practicing more number of PYQs is very important, and knowing previous year question paper is very important. Okay, pattern is very important. So you can check out the NTA PYQ where we have been segregated all the questions, previous year questions together, chapter wise, topic wise, and all. And you also have a Brahmastra test series. So yeah, and a light plan also. So go check out and let us know if you're feeling any difficulties with the topic. Thank you.